Hello and welcome to lecture number 21. Today's topic is mutation and DNA repair. So last time we discussed genomes and we just sort of looked at them holistically, what a genome is and how you sequence it and what kinds of sequences there are and how you can compare genomes with each other and so forth. So specifically we said that whole genomes can be sequenced and assembled. I showed you a little bit of the algorithm that's used to assemble genomes. I said that genomes are made up of many types of sequences, and we looked at some of those types of sequences, such as repeat regions, transposons, satellite repeats, introns, and so forth. And then we discussed the fact that computers can be used to look for telltale sequence motifs. So motifs, in other words, little stretches of sequence that are specific and unique. Well, unique in that they code for something specific, but they can be found in various places throughout the genome and can therefore be used to predict the function of a stretch of DNA, such as pieces of in the promoter region for recruiting transcription factors or splicing junctions between exons and introns. I talked a little bit about genome comparison using trees and looking at evolutionary relationship. We talked about polyploidy, the whole genome duplication, and then we looked at compacting of the genome in eukaryotes versus prokaryotes. And then at the very end, we talked a little bit about viruses. So today we will talk about mutations and how mutations arise and how frequently they arise, and then also how these mutations can be repaired and how frequently they get repaired. All right, so your book starts with these two graphs, and I want to talk a little bit about those two graphs. They're interesting. They're a little bit confusing in some ways, but I think your book also describes quite well in the text what to make of these graphs. So spontaneous mutations are the results of mistakes made by the DNA polymerase during the DNA replication process. Of course, they can also occur due to chemicals or UV radiation, things like that, so mutagens. But they can also just simply spontaneously occur without being exposed to mutagenic substances or UV light. So what you see on this graph is the rate of mutation, so how frequently mutations occur, per nucleotide pair, so per site essentially on a genome, per round of replication. So this is on each site, how frequently, how likely is it that that particular nucleotide will be, there will be a mistake. And there seems to be a relationship between the frequency with which there are mutations, in other words, with which there are mistakes made by the DNA polymerase, and the genome size, so how big the genome is. And we talked about genome sizes last time, and we saw that genome sizes of bacteria and viruses are small, so they range from 10 to the 4th to 10 to the 7th base pairs, while larger, more complex organisms, including insects and vertebrates and yeast and worms are much larger genome sizes. And so it appears that the larger the genome size, the lower the mutation rate per nucleotide. And so what I say here on the side is that spontaneous mutations are not the result of mutagens. I say that mutagens increase the rate of mutations. In addition to that, that virus polymerase seems to make mistakes only every 10,000 to 100,000 base pairs, but that polymerases in humans and mice and other cellular organisms here, multicellular organisms here, seem to make the mistakes much less frequent. So that said, so it appears that the polymerase is just more faithful. It's better in the mice and humans and multicellular organisms compared to viruses. And that is true because the polymerase has proofreading function. But if you now look at, it has proofreading function in the multicellular organisms, but not in the viruses. Now, if you look at the rate of mutation per genome per generation, this is now per entire genome per generation. So in other words, not just on the per nucleotide basis, but if you have a larger genome, then you make more mistakes per generation. So this is now all the cellular replications that occur from one generation to the next. If the generation is just one cell because it's a single cell organism, then you would assume that there are fewer mutations made overall than if it's a multicellular organism that has many cells that duplicate many times during the lifetime of an organism. And so that's why now humans and mice and fruit flies are so much higher here at the rate of mutations as opposed to viruses and bacteria that are down here. So essentially what that means is that a human F1 passes about 30 new mutations to the next generation, so to the F2 generation, that they did not already inherit from their parents. So per generation, 30 new mutations are made and then passed on to the next generation, potentially passed on to the next generation. Humans have a higher absolute number of mutations per genome than bacteria, despite the lower mutation rate, because of the larger number of cells and the nucleotides per genome. So I said potentially passed on to the next generation because they would have to be in the part of the genome that gets packaged into the gametes and then sent off to the next generation as egg or sperm. An important other thing to remember is that the parts of the genome that are actually genic, in other words, that are coding for proteins, are only a few percent, right? You saw that on the pie chart for the humans last time, it was maybe 5% or so. So 30 new mutations anywhere in the genome may not actually affect many protein genes. So that's another thing to remember. Okay, so with respect to what I just said, that cells that are not passed down to the next generation because they're not germ cells are called the body cells. And so body cells are cells that aren't going to be passed on to the next generation. They stay within the same individual. And so here's an example for somatic mutations that will not be passed down to the next generation of this morning glory plant. So each sector, each blue sector that you see here, is the result of a mutation, and that mutation allowed the petal, the tissue here, to produce a blue color. And when it happened in one cell, that mutation was passed on to the next cell through mitosis. And so therefore you see these sectors where one mutation occurred maybe right here, and then all of these cells back here from here on out were affected. You can see here that there was a mutation in the cell that gave rise to this entire sector. So each blue sector on this flower arose from a single mutation in a single cell, and it was passed on to the daughter cells of that cell that had the mutation through mitosis. So in this example, the same particular gene was mutated 11 times, creating these blue sectors. Now that doesn't happen very frequently, that the same gene is mutated so many times. That is a feature here of this morning glory. You'll learn a little bit more about that later, why this and why particularly this particular same gene was mutated so many times. But of course, that is what gives the morning glory, this particular morning glory, some horticultural value. But the point that I'm trying to make is that these are somatic cells. These are not going to be passed on to the next generation. And that once a mutation is made, then the offspring cells, somatic cells, body cells, not germ cells, will also carry that mutation. Now, if we switch our topic here from plants, looking at plant somatic cells, to animal or maybe human somatic cell mutations, there is, of course, the example of cancer. Cancer is a mutation. It arises when in a cell a certain number, it's assumed that it's usually about 3 times, certain genes are mutated in the same cell that then lead to unregulated cell growth. We talked about that in a previous lecture. We talked about the proto-oncogenes, tumor suppressor genes that were regulating the speed with which the cell cycle goes forward. So the point of this slide here, in this context of the lecture, is that multiple mutations in several of these cancer genes are what is required for a cell to become cancerous with respect to malignancy. So the slide here shows normal cells, and the cells, they divide, and then they will produce another cell, and that will divide, and it's normal every time. With the exception of this cell, where a spontaneous mutation, or maybe a mutation through a mutagen, arose in a particular gene. This is the APC gene. So that is a gene that oftentimes, when mutated, can give rise to cancer. The same as with the RAS gene and the P53 gene. These two we discussed briefly when we discussed those two types of genes, the tumor suppressor and the oncogenes, with respect to cell cycle regulation. Anyway, so this particular gene here, a mutation arose, and so from the normal cell, we now start to see some slight changes in the anatomy of this particular cell, or the cell type that arises from many of these cells that are now all blue, because all of these blue cells carry that mutation in the APC gene. And so from that, a bunch of cells will now form, and they form a small benign polyp. So this is not cancerous, but it's growing a little faster. So now we have a second mutation in the same cell that in which the APC gene was mutated. And let's assume this is a mutation in the RAS gene. That was one of those signaling genes. So now these cells, of course, all give rise to cells through mitosis that have both of the mutations, the APC and the RAS gene mutation. And cells that grow like that now produce larger, still benign polyps. So those are larger cell masses. And now if there's a third mutation, let's say in the P53 gene, that can then cause all of its offspring cells through mitosis to be cancerous cells, and that can then lead to the formation of a malignant tumor. So in this case, colon cancer, for example. So what I'm trying to say here is that mutation can arise. It can be passed down to the next somatic cell, and that in the case of cancer, many mutations in the same cell can then lead to the formation of cancerous tissue, of tumors, malign tumors, and so forth, malignant tumors. The risk factor that you sometimes hear about when you hear about people with certain disease risk factors has to do with inherited mutations that already come inherited to the person with the risk factors. So the risk factor is, for example, already having been born with a mutation in one of these genes, so that the accumulation of only 2 additional genes would then lead to the formation of cancer. And so therefore a genetic risk factor, such as an inherited mutation in a cell cycle regulation gene, reduces the need for additional somatic mutations for a cell to become cancerous, right? So if you are already born with a mutation in APC, then that is a risk factor for cancer. It doesn't mean that you get cancer, because this may never happen. They might all just be blue. You might be more prone to having certain polyps, but they're not cancerous necessarily. And so that's what a risk factor means for, in this case, for cancer. Okay, so now let's take a look at the types of mutations that can occur in the genome and how we classify them and how we discuss them when we talk about mutants and mutations. So there are small mutations called point mutations, if they're just occurring in a particular nucleotide, or there are larger mutations where a chunk may be missing, or where a really large chunk may be lost or placed somewhere else in the genome. So we'll talk about all of these from the small to the large during this lecture. So let's start with the point mutations. Point mutations are fixed. That means made permanent, not repaired. That's not what this means in this context. Made permanent in the second round of replication. I want to show you why that is. So if this is a piece of DNA that is going through replication, then both of the strands are being copied. Now, let's assume there's an erroneous incorporation into one of the daughter strands, where you, instead of an A, where the polymerase put in a G. And here it put in the correct T. So there's no mutation on this strand, but on this strand there is a mutation. Now, before this mutation gets fixed, before it gets made permanent, there is a chance, there's time for the cell, for the machinery of the cell, to repair this piece. And at this point, it's possible to detect that because these two don't really fit together. So if that was undetected, so if the repair machinery didn't work, then after the second round of replication, then you have the fixation of this mutation. So now then the polymerase will go here and look for a T, and it will put in an A here, but it will look here in the other direction, and it will say, oh, there's a G, so therefore in my new copy I have to put in a C. And at that point, there's no obvious mistake anymore that an enzyme could notice that they just don't fit together, because now they do fit together, because now both of these nucleotides have been changed. So then there is a change from a TA to a CG. That is called the fixation of a mutation, but that would require 2 rounds of replication. So during that time, there's time for repair. We'll talk a little bit about repair mechanisms at the end of the lecture. So let's first see what kind of mutations can arise and what their consequences may be. So there are 3 types that your book discusses, and so let's it uses different types of terminology. I want to point that out for you so that you don't get confused with it. All right, so we have so-called synonymous mutations and non-synonymous mutations, and then here we have an example of the non-mutant. Right. So let's say here's a piece of DNA, and if you look at this DNA, there's a mutation here, and there's a mutation here. And these 2 mutations are both point mutations, but they seem to be different because this one's called a synonymous mutation. This is a non-synonymous mutation. Why are these 2 mutations different? So in this example, this gene codes for these particular amino acids, and this is happens to be a stretch of beta globin. So this is a protein. And after transcription, you get the RNA, and then after the RNA is translated, you get the protein. And you see that we have, you could look it up in the table, right, with the codon, with the coding table, where you see the codons, and where you can see the amino acids that go with each triplet. You could see that CCU stands for proline, and GAG stands for glutamine, and there's another glutamine here. Now, if you have a change from a G to an A, then GAG and GAA both code for glutamine because of the wobble. Remember the wobble? That is the third amino acid, the third nucleotide position that doesn't often change what the triplet codes with respect to the amino acid. So whether it's a GAG or a GAA, both of those triplets code for glutamine. So therefore, it doesn't matter, if you will, for the protein, because there's no change. So therefore, it's called a synonymous mutation because it sort of means the same thing. Right? Synonymous means the same, meaning the same meaning. And the GAA here has the same meaning as the GAG, even though it is a point mutation. So that is a synonymous mutation. If it's not the same, then it's called a non-synonymous mutation. And so here you have a case where the second nucleotide in the codon triplet is mutated. And if you change the GAG to a GUG, then you do change the amino acid that it codes for from glutamine to valine. So a nucleotide substitution that changes the amino acid is a non-synonymous mutation, and that changes the protein sequence. Now, see that there are two terms that are the same for these terms. So a synonymous mutation is also referred to as a silent mutation. That makes sense, right? Silent, it doesn't you don't see any difference. And this is a missense mutation. So it still makes sense, but it's not the same sense. It makes a different type of sense. It's a missense mutation. And then there's a third type, and the third type is over here. And so here's our non-mutant example, and that is when a nonsense mutation occurs. And a nonsense mutation means it makes no sense in terms of adding a different amino acid. It just stops the translation process altogether. So if you have a mutation that changes a codon that is coding for an amino acid to a codon that codes for stop, for the stop codon, then you get a premature stop in the protein, and that is a truncated protein, much smaller protein, depending on where that mutation occurs. If it occurs very early in the protein, then you just have a few amino acids and they have absolutely no function, and the whole thing is completely useless. You might get a stop codon very late in the process. By chance, there might be a mutation only 1 or 2 amino acids before the end, and that could be, doesn't have to be, but it could be in a region of the protein that is not so important, and so therefore the mutation may actually not affect the function so much, but it could. So we have 3 different types of point mutations. We have the silent mutation, we have the missense or the non-synonymous mutation, and we have the nonsense mutation. So those 3 types of mutations are point mutations, where we have the terminology for nonsense, missense, or the non-synonymous, and the silent or the synonymous mutation. So those 3 types with those 5 terms you should know. All right, let's take a look at what happens when other types of mutations occur. So you could have a chunk missing or a larger piece missing than just it change a point mutation from one nucleotide to another nucleotide. And so there are 2 examples here. On the left-hand side, we have an example that I go through in a second, where we have the loss of a single amino acid. And here on the right-hand side, we have what's called a shifting reading frame. So that is when when there's an insertion of a nucleotide that wasn't there before, when that then messes up the triplet code, because now you're not reading 1, 2, 3, 1, 2, 3, but you're reading maybe 2, 3, 4, 2, 3, 4, 2, 3, 4, which of course means something completely different in the code of the amino acids that they stand for. So let's go through these left and right one at a time. So this is an example of a transporter. So this is a chloride ion and chloride ion channel, and this particular gene, this CFTR gene, codes for this. If it's expressed, if it's transcribed normally, then it gets translated into this sequence: isoleucine, phenylalanine, and glycine. Now, there is a mutation known in the CFTR gene where these 3 nucleotides are missing. The whole set of 3 nucleotides are missing. And so what's missing here is the middle amino acid. So it goes straight from isoleucine to glycine. And in the most common mutant proteins, 3 nucleotides are deleted. This is the mutant protein. This is the most common type in this particular gene, resulting in a missing amino acid at position 508. And remember that this goes on over to the left and over to the right, right? So that's why this is position 508. We're just not showing positions 1 through 506. So if it's normal, that protein gets made, and it's a protein channel that pumps these chloride ions out of the cell. But interestingly, and that's not a rule, but in this particular case, in this particular protein, the lack of just a single amino acid can make the protein unstable, and then it gets degraded and it's gone. So it never makes it into the membrane. And so therefore, people with this mutation are missing this CFTR transporter, which leads to disease. Now, I said earlier, in some cases, in other cases, different genes, different positions of genes, a different amino acid or a missing amino acid may not change the function, or it may only marginally affect the function. But it could certainly affect the function depending on whether it's an important amino acid, and whether or not one is important, we will only know when we look at the function, or we look at the three-dimensionality, how the protein folds, and how it has to be held together so that it's stable. And in this case, missing that particular amino acid leads to complete instability, and so that protein is completely gone. All right, so that is a loss, a deletion of three amino acids, three nucleotides leading to an amino acid change. Here in this case, we have an insertion. So here's a deletion, here's an insertion. An insertion can occur when there's a stalling, or when there's an extra nucleotide gets put in on one side but not on the other, and then in the next round that change gets fixed, right? Gets made permanent. Then you have an insertion, and so here you see this particular gene, and then you see that there's an insertion here, and you can see that instead of CCU coding for proline and GAG coding for glutamine, you now have CCU, and then the GAG over here, right, that is this GAG, that's still there, but it's not coding for glutamine anymore because there's this extra C. And so with that extra C, the translation machinery will look at it as the beginning of the next triplet. So it will then say CGA. And CGA does not stand for glutamine, that stands for arginine. And then, of course, from there on out, all of these triplets are now shifted one over. And so I write here, shifting the reading frame where three nucleotides code for one amino acid by only a single nucleotide causes all following amino acids to be wrong, right? So they're all different. Here we have glutamine, lysine, serine, alanine, but here we have completely different ones. And so this will make something that it might be a protein but has no function that we know of. And so therefore, the function of this protein is lost because of that insertion of a single nucleotide leading to a frame shift mutation. Now you could insert three nucleotides. I mean, this could happen, right? That three nucleotides potentially are inserted, or 6 or 15, a number of 3. And that might insert amino acids that could, just like in this example over here, make that protein unstable, and it gets degraded and it's completely gone. But potentially, it could also it keeps the reading frame intact. It just adds an amino acid in between. And that can be detrimental, but it doesn't always have to be detrimental. That depends on the individual mutation and the three-dimensionality of the protein structure that is affected. So here we have not point mutations, as I showed you in the 3 examples before, but now we have insertion and deletion mutations. Sometimes they're referred to as indels, insertion and deletions, indels. And that is a type of mutation as well. All right, so now let's look at a mutation that we discussed a little bit last time, and we didn't really refer to it as a mutation. We just referred to it as a feature in genomes, and we said that these particular sequences that are called transposons are frequent, and you saw that retrotransposons, in fact, make up almost 45% of the human genome. They're so frequent because they copy themselves and insert themselves elsewhere, right? That was this copy and paste mechanism of a stretch of DNA that can make a copy of itself and then insert itself somewhere else. Now, today, I want to discuss transposons from a different point of view, not just as a bunch of DNA that's taking up space, but what happens when a piece of DNA jumps around. And so we said there were two types of transposons. There were the DNA transposons and the RNA transposons, or the DNA and the retrotransposons, which are RNA transposons. And the reason why the retrotransposons are called RNA transposons is because they make a copy of themselves by going through an RNA intermediate, and then that RNA intermediate is back translated or back transcribed into really into DNA, and that is then inserted. And then there are the other kinds that are pieces of DNA that excise and jump. So that's the difference between the copy and paste versus the copy and paste. So copy and paste and cut and paste is what I mean. So cut and paste versus copy and paste. So here's an example of a cut and paste transposon and how that can act as a mutagen in the cell. So you don't need to be exposed to mutagens because you have those in your cells at all times. All right, so let's look at the slide here. This is an example from corn, and I mentioned Barbara McClintock to you in the last lecture, who won the Nobel Prize for finding out about transposons and transposition within genomes. So she used corn as an example as her study organism to learn about this. All right, your book doesn't explain this in much detail, but I want to add a little bit so that you appreciate this. These phenomenally interesting little pieces of DNA. So here on the bottom, let's start with the bottom actually. We have a blue gene. This is the blue gene, and it's uninterrupted. It's just a normal gene. It's making its protein, and it's functional. And what it's doing is it makes a purple color that you can see on these corn kernels. So these are corn cobs. And you see that these kernels are purple, and it's because this particular gene makes that purple color. At some point, a transposon might be active, so a piece of DNA that cuts itself out somewhere and then jumps elsewhere in a random place in the genome. And when that happens to land in a gene, then it disrupts that gene. So that gene is now interrupted. And when it's interrupted, it's most likely not functional anymore because there's so many pieces of nucleotides now in between that the reading frame is off, right? So you have changed the reading frame, producing basically some garbage protein that the cell doesn't need or doesn't know how to use. So in the example up here, a transposable element, that red bit here, was inserted itself into this blue gene. So I write up here, the blue gene is interrupted by the red transposon. The interruption makes the purple color gene non-functional until, and only then, until the transposon leaves again, causing purple sectors. Okay, so now let's think about this from the point of view that you have a chromosome here with this particular purple gene that's disrupted and not functional. And so if you now look at the corn, you see all these kernels that are yellow, and that's because you see the layer underneath the purple color. So there's purple on top of it, a purple layer on top of the yellow. But that's not functioning in these yellow kernels because the red transposon sits in between there, it disrupts the gene and it makes it unfunctional. But when the transposon jumps out into a different location, different chromosome here, it's just anywhere else. Could also jump over here, it could jump over here. Let's say in this case it jumps into this purple or pink or whatever the other color here is, chromosome, and it leaves that gene that it previously interrupted. It can reinstate the function. So this gene is now functional again because the mutation has left. So this is basically an example. This is an example of unmutating a gene. And when that happens, then let's say there's a cell where the transposon leaves and unmutates the purple gene, then you get a little sector, just like on the morning glory picture before, you get a little sector of tissue where that occurred and where now the purple gene starts to produce purple pigment again. And so every bit that is now purple is because there's a specific event that occurred where the transposon jumped out of the gene and made the gene functional again. And that is actually the case also with those somatic mutations that I showed you over in the morning glory example. But your book doesn't specify that. So when the transposon leaves, the transposable element is now excised, the gene is now functional, and you can see these sectors arising. And so in this case, there's just, all of them are okay. In this case, you have a set of them that are completely okay, and some of them where in some parts of the tissue cells underwent the leaving of the transposon and therefore the return of the purple color. So imagine thinking about this or looking at crosses. This was all genetic work that Barbara McClintock did, looking at this and then deciding, it does not really follow my Mendelian genetics that I know, therefore it must be something different. And so therefore, let me come up with a different hypothesis, which is essentially what she did. It's just mind-boggling how she could think that this might be the case, and then she followed it up with a lot of other experiments, and then when it was ironclad, then published it, and eventually got the Nobel Prize for that. So transposons are jumping genes that can cause mutations. They can actually also unmutate certain genes. And I'm only using the term unmute. That's not something you'll find in the book. Okay, so we're talking about mutations, right? We're talking about types of mutations. We now looked at point mutations. We looked at indels, or insertions and deletions. We looked at transposons in the previous slide. And now we're going to look at deletions and inversions and translocations. And so what I'm showing you here is that we're going from the small to the large, right? So now we have large sections of chromosomes that can accidentally be rearranged, becoming very large mutations. So this shows a normal chromosome with this particular stretch of DNA. And as it appears from sequencing chromosomes from different organisms, individuals of the certain species, it has been found that there are chromosomes, this chromosome, that can also be found to have a duplication of that region. How strange, right? So that entire piece was duplicated somehow, and you have a large stretch, could be thousands of base pairs, could be many more than thousands, tens of thousands of base pairs that are duplicated, and where maybe other individuals in that population have a deletion of this piece. So somehow this duplicated, but then deleted on the other chromosome. Maybe one became the chromosome that moved into one part of the poles during meiosis and the other one to the other one. That's a possibility. And I'll show you how this works, or how it could work in one example in a little bit using an animation. So for right now, I want to just explain what a duplication or deletion is. So large-scale deletion and duplication. An inversion is a piece of DNA where the direction of the sequence was flipped. So basically, this example here was just so you can see the inversion, where the piece of DNA was essentially cut here and here at the end of the red and the beginning of the green, flipped around, and then back inserted into the chromosomes. And that is called an inversion. So you can compare the normal chromosome individuals with a normal chromosome and individuals that have the inverted region of that chromosome. And then you can get even things like translocations, where translocations are essentially breaks of chromosomes where one piece of the chromosome can fuse with the other part of a different chromosome and vice versa. Now, this looks sort of like crossing over, right? This is not crossing over because these are not homologous chromosomes. So this can happen at other times of the cell cycle, where the chromosome breaks, maybe due to mutations, maybe due to UV radiation, or maybe due to gamma radiation, radioactivity, and so forth. And when chromosomes break, there are repair mechanisms, which we will discuss just a little bit at the end. And those repair mechanisms can then fix the broken chromosomes, but sometimes they fix them opposite. So they the repair mechanism doesn't know that this goes here and this goes here. It just knows that there are pieces that need to be stuck together. And so it might stick this together here and that together there. And so that leads to translocations of large portions of chromosomes. So now you don't have a loss of the DNA, but you have a different location where that DNA or that particular gene is. So I find this difficult to grasp. How does this even work? And why does it work? And how does this happen in the cell? And a few years ago, I came across this particular website that I find absolutely stunning. And I'm going to show you a few parts of it. It has some animations, but I have the whole website here for you, so you can go back to that later on if you want to see more. So let me find where it is, and then show you some of those. So here's the website that I mentioned. Let me give you a little bit of context. These people that put this website together, they are researchers, they're transposon researchers. And they have, over the years, many, many years, they have sequenced many different individuals. I think this is mostly done in corn. So they've sequenced them, and they know what the unmutated, the normal, the wild type, what that sequence should look like. And so then they can compare the sequence of odd ones, of mutants, with the wild type sequence. And then from that, they reconstructed how the change must have occurred. And that is what they're animating. So there's one thing that I didn't tell you, and your book doesn't discuss at this point, because transposons were introduced not so much with respect to how do they do it, but what are they? So how do transposons, how do they make the cut and paste mechanism happen? There's one thing that I can tell you about, and that's easy to understand, I think. That is that in that stretch, let's say this is a transposon here, this red bit. It encodes its own enzyme that does the cutting and pasting. So it's called a transposase. It's an enzyme, it's a restriction enzyme really, that recognizes its own ends and then cuts itself out. It does all of that and then helps it insert itself somewhere else randomly. And you will see these little blue dots here in the animation. And the animation uses these blue dots to indicate the transposase that is being produced by the transposon itself. But that's not what I want to show you. I want to show you how these types of large-scale mutations can arise because of transposons. So look at this. So here's our transposon. These are the transposases. These are exons. This is a particular gene. Doesn't matter where it is. Happens to be the centromere, happens to be here. And this is the target site according to this graph. Now, the target site is only known in retrospect. So as I said, they looked at mutants, they sequenced them, they knew that this transposon later on was found here. And so therefore, they called it the target site even in the unmutated version. So I'll show you the unmutated version, which is this. And then I'll click that button and you see how the transposition causes the change in the location of the transposon and therefore change of mutation within that chromosome. Okay, so the blue balls are the transposases that were made by the transposon itself, and now you see that they help it move somewhere, and then they just happen to land there at this spot AB, and it inserts there. So now this part here was unmutated, as I call it, and this stretch between maybe gene A and B was mutated. And if this is a gene, then that gene is now interrupted and potentially no longer functional. Right? So far so good. That should be relatively straightforward. Now let's look at how all kinds of weird mutations can occur because of transposons. So this is another example. These are real examples, right? This is what was found, and then how the animation explains how it must have occurred. First of all, see that there are multiple types of transposons here. I'll start it. This is already the end configuration. So this is how it starts. This is how it starts. So we have the transposons here. The chromosomes look great, right? So here's the transposase. Let's get it started. So here you see that the transposase cut, but it cut on the two different sister chromatids. And see, now we have an inversion of a piece that previously, I'll put this back up so we can see that again. So here's AB, right? That piece is now inserted over here. That makes that arm much longer. The bottom arm much longer. And you see that this piece B is now also found over here. So now you have an inversion because of the transposon, because the way that the transposase inserted this transposon. I'm not expecting that you remember and that you can explain that and that you can show that or that you can verbalize it even, but I just want you to show how these weird kinds of rearrangements can occur. Here's another example. So which one did I want to show you? This one here. So here, two different adjacent transposons don't excise completely, but lead to a change in the way that the DNA is assembled, right? So we'll do this again. You see C, D, and E, those are the locations of this particular gene or genomic location in between these two transposons. Now transposase is expressed from within these transposons, happens to bind to the two different transposons ends, doesn't fully cut it out, but changes the position of the DNA, and now you have E, C, D. So now there's a change in the way that the transposon, that the DNA is now located. Let's look at this one here. So here we have transposons on one chromosome and no transposon on the other chromosome. So if you happen to have excision of this piece and activity of the transposons here, then if they bind to different, if they coming from one chromosome bind to a different chromosome, do the cutting there and the insertion, but since there are two pieces, lead to a translocation. Now we have the green arm on this chromosome and the black arm on the other chromosome. So you can come back to this particular website. It's up here. You can read a lot more about transposons and what kind of weird things they can do. But all I hope that you appreciate from this little bit of discussion here is how transposons can lead to all these different types of deletions or inversions or translocations that can happen within the genome. Now, this can be completely detrimental, and so therefore that that organism might be taken out of the gene pool because it's not viable, that kind of damage. But if it is viable, then there's large genomic change that occurs, and that can be potentially of environmental, of evolutionary importance, because maybe now genes are in a different arrangement or the transcription is different, maybe environmental effects are differently perceived and responded to by an organism. That's basically how evolution works. So this can happen, and I showed you one way how these types of large-scale mutations can occur. All right, let's spend the last few minutes about DNA damage and DNA repair. So DNA damage can lead to mutations, but it doesn't always have to lead to mutations because we have repair mechanisms. Organisms are all the time exposed to mutagens. Now, they could be exposed to chemical mutagens. Those may or may not be found in nature. So there are natural mutagens and there are man-made mutagens, carcinogenic products or compounds. But there's also UV light, so we're always exposed to UV radiation, and so there's mutations that can occur that way. But then I also showed you about the spontaneous mutations. So all of these mutations, or at least many of those mutations, can be fixed by repair mechanisms. And we'll talk about those repair mechanisms. But let's first look at the types of damages. Here we have a single-stranded break. Here we have a missing base. Here we have a double-stranded break. Those are particularly detrimental because it's difficult for the enzymes to know, which ends go together if they join them back together. The bulky site group that's attached to a base. So this is basically some sort of a mutagenic compound, a chemical compound that just happens to insert itself between the DNA. Some such compounds exist, and so those are carcinogenic. There's also cross-linked thymine bases, so two T's next to each other. They have the propensity of forming this TT bond and basically breaking the hydrogen bonds in between their pairing partners when there's just enough energy that shines on them from UV radiation. So you get a lot of thymine-based cross-links when there's a lot of UV damage. And so this can be repaired, but that can also lead to the DNA polymerase, when it comes along here to make a new copy of it, stalling and not seeing that these are two T's, but reading them as something else. And then in the new nucleotide strand, putting in maybe too many or too few nucleotides. So that can happen because of these cross-linked thymine bases. Other bases can also cross-link. Okay, so there are three repair mechanisms that I'll go over with. They are relatively similar in some cases, or in some ways. They reuse a number of enzymes, but they have some specific enzymes that they also use. So the first one of these three types of DNA repair mechanisms that we'll discuss in this class are the post-replication mismatch repair. And the post-replication mismatch repair is just like the name suggests. After DNA replication, there may be a mismatched nucleotide. And so I say here, the reason for that mutation is that the DNA polymerase makes a mistake that it doesn't fix during replication. And so now a set of proteins are dispatched by the cell. And these different types of proteins, they bind in certain places. And we don't have to look at all the exact details. You'll learn more about this in genetics or in molecular biology in those classes. But what I do want to point out for you to learn here in Bio 111 is that the set of proteins can recognize this little bump here, right? There's something off with this TG because it shouldn't exist. It should be a TA or a GC. And so proteins can recognize that. And when they bind, when they're recruited, they eventually get an enzyme to work that's called an exonuclease. An exonuclease means it cuts away. Nuclease is anything that cuts DNA. And an exonuclease cuts something out, cuts from the end away. So there's a little break that's made, and then the exonuclease cuts off a stretch more than it needs to around this lesion, around this mistake. And then at the end, there's an enzyme that comes in after the nuclease has it removed, and that's the polymerase, the DNA polymerase, that then puts in the new nucleotides, basically tries again, right? So it does what it did before, but it messed it up and it didn't repair it right away. And so now we go at it again. We cut out the wrong piece, a little bit more than that, and then have the DNA polymerase try again. And it's pretty good normally, right? So it makes mistakes very infrequently. So second time through, it's most likely correct. So the DNA polymerase fills in these missing nucleotides. And then there's an enzyme that's called ligase. We mentioned that before earlier in an earlier lecture. Ligase always binds together two nucleotides, basically joins those pieces together. We discussed that in the context of DNA replication. So The reason for why this particular set of proteins is dispatched is because the polymerase made a mistake, and so now that has to be fixed. Certain proteins are employed. Ligase is one of them. Polymerase is one of them. And so if you know that much, I think that'll be enough for Bio 111. A different type of DNA repair mechanism has a similar set of enzymes. You see a ligase in the end again, but it uses a certain different set of proteins that recognize that kind of a mistake. And so that kind of mistake, that the process that's called base excision repair recognizes, is when a uracil, so U instead of a C, is found. So the U shouldn't be in there in DNA, right? It should be only in RNA. But chemicals can affect, certain kind of chemicals can affect other kinds of chemicals, right? Chemical reactions, essentially. So carcinogens can be carcinogenic because they mess with the structure, the chemical structure of the DNA itself. And when that happens, that can happen relatively frequently to cytosine, where you have deamination, so loss of the amino group here, and replacement with oxygen. And so you see that this is almost the same. It has the hydrogen here, but the amino group is gone, and there's this oxygen, double bound oxygen over here. There's an enzyme that can recognize that this is uracil, this is cytosine. So when this particular enzyme recognizes uracil built into the context of DNA, it notices that something is up. And so this enzyme called DNA uracil glycosylase that will cleave the sugar backbone over here, remove the uracil, and then an enzyme will cut out the entire. So first it makes a cut here, then the entire piece gets removed by another enzyme, and then a third enzyme comes in. There's always DNA polymerase needed that then fills in the this little gap, and then ligase always seals it up. So you see here that I write, the reason for the mutation is that the DNA polymerase either inserted uracil by mistake or cytosine was converted by chemicals to uracil. And now we have a set of enzymes that will fix this, ligase always being one of them at the end to complete the repair. And then the last one that we'll discuss, that's not the last one that exists, but the last one that we'll discuss in Bio 111 is the base excision repair. So base excision repair is also relatively similar. Base excision repair can, for example, repair thymine dimers or a stretch of damaged DNA where chemicals have somehow damaged chemically the DNA, other than in the uracil example. So, for example, these two T's that I mentioned earlier that then mislead the polymerase and stick in something other than two A's in there. So that can be recognized by the base excision repair system. The base excision repair system, again, is a bunch of enzymes. Some enzymes we've already heard about. The ligase, always at the end, seals the ends together. There's always a polymerase that sticks in the new pieces that have to be replaced. And then there are some enzymes that recognize the mutation, and there's some enzymes that cut parts of the DNA out so that the new piece can be filled in. So it's sort of a little bit like a dentist. It finds a hole in your tooth, and it doesn't just fill that hole, but first drills out a whole large portion, or relatively large portion of it, and then fills that in with something new, and then seals it up so that it will stick and that it will fit and seal that hole. So that's sort of the process by which this works, except that for base excision repair, the reason for the mutation here is DNA damage, for example, by UV light, and then it recognizes specific types of mutations and then employs its specific proteins that recognize, cut out the mistake, and then take DNA polymerase and DNA ligase to fill these strands. And so that is how DNA gets repaired, and that is good that we have so many different ways of repairing DNA, because mutations occur all the time, and if they weren't fixed for the most part, then proteins would be losing functions frequently, and there would be a lot more loss of viability in the next generations. So let's summarize what we discussed today. I said that mutations occur spontaneously and randomly. We said that only mutations in germlines that make it so that the gametes, the germline produces the gametes, the line of cells that is giving rise to gametes, are passed to the next generation, and that we have somatic cells that can also sustain mutations, but they're not passed on to the next generation. But they might be just as important, especially for that individual, with respect to changes in that organism during its lifetime. And cancer was one of those examples. We talked about different types of mutations. I talked about silent mutations, which are also called synonymous mutations, the missense or non-synonymous mutations, and nonsense mutations that are distinguished and that I suggested that you know. Deletions and insertions. We talked about transposons a little more in detail this time, and I showed you how they can translocate by inserting and excising and thereby mutating or reactivating genes when they leave the place where they originally mutated a gene. And then I showed you this website where we looked at how transposons can be the reason for these large chromosomal changes, such as duplications, inversions, and translocations. In the end, we spoke a little bit about DNA damage and how DNA polymerase can make mistakes and how that can be repaired by certain repair mechanisms. And that is it for today, and I will see you in class.